Hello, my name is Jalen Furqan. I go to Morehouse College. This summer, I got to go to Natick Soldier Center, and I got to work in the Skin Microbiome lab with my mentor, Jordan Whitman, and this is the project we worked on. We worked on development of a simplified microbial community of the skin microbiome. So a little introduction, in the skin microbiome, there are thousands of different bacteria that lives in the skin microbiome. It's also diverse, composed of different viruses, fungi, and bacteria. So it's hard to study all this bacteria at once. So a goal or objective of our lab is to create a simplified, simulated version of the skin microbiome. This will allow us to study the community dynamics in a co-culture environment. And like our future goal of doing this is to integrate these simulated versions of the skin microbiome with organ-on-a-chip technology, or MPS chips, microphysiological chips. And what is organ-on-a-chip technology? It's a lab techniques to help you study host biology and recreate main body parts. So being able to do this will help us make different interacting cell lines, realistic microenvironments, and different realistic continuous fluid movement, such as like blood and sweat. And once we integrate these two mock communities and this organ-on-chip technology, we'd be able to study how the tissues respond in the simulated skin micro community, and we'd be able to add real skin microbes to the simulated skin community, and then we'd be able to simulate real-time infections, healing, or different exposure outcomes, and we can see how the skin microbiome can be harmed or how it can help the host. So here's our methods. We did a 9-member co-culture and mock community build. You can see our 9 organisms here, and you can see their roles and functions. Some organisms I want to highlight is S. epidermis, which is a commercial bacteria that helps protect the skin and hydrates the skin. But if it's cut or wounded, it can harm the skin. And another organism I want to recommend is S. furfur, which is the only fungi organism on our co-culture. So what makes our research different from previous studies, and they only study bacteria, but we're trying to be a little more diverse by studying the other components in the skin microbiome, such as fungal in our organism. So in our methods, we did an individual co-culture study. So we made a control environment to study these microorganisms' behavior, their different growth patterns, survival rates, and their colonization behavior, and then we can mimic the pH and the temperature in the skin. So we also had to find the appropriate media for these organisms in our mock community builds in our co-culture to grow. So we did media optimization, and we had 2 experiment approaches. Our 2 experiment approaches was the 9-member physiological proportion. This mirrors the abundance of natural human skin. And then we did our 9-member balance, was we put all the organisms in the co-culture at an equal amount for competitive dynamics. So we wanted to find the growth patterns for these 9 organisms in the co-culture. So we did qPCR and this will help us track the abundance and how much of the organisms are in the co-culture. It helps us track how it grew, how these microorganisms grew, survived, or died out over time. So here's a little experimental workflow and see what we did in the lab, our experiments, and how the order of operation, how it worked. In our predictive findings, back to our 2 community approaches, we did balance, which is equal amount for competitive advantage. And then we want to know physiological, which is real life skin, mimic real life skin. So if you look at our figure here, S. epidermis, this is the organisms that grew in our co-culture. This represents the CFU count of that organism at different time periods, showing how it grew in the co-culture at different time periods. And you can see our 2 our 2 community approaches, balance and physiological, and you can see how it didn't matter what approach we took, it grew no matter what. So it's just representing how stable this organism was in our co-culture environment. So our predicted Army impact, why is this important? Our goal is to integrate these mock communities with the organ-on-a-chip system, for we can make a realistic skin community. And this will allow us to make models for different future exposure studies in a lab environment. This will allow us to study how the microbes and tissues respond to biological threats. And then we can also do environmental stressor simulations and different host microbiome dynamic simulations. And then ultimately, this will help us design antimicrobial treatments, different root care strategies, and make protective gear guided by skin microbiome research. Thank you.